Okay, so I welcome everybody. Thank you for joining in for this rendezvous. Today we have with us Dr. Vaishnavi Ananthanarayanan, and I would invite Sanskriti to start with the introduction of our speaker.
Hello everyone. Dr. Vaishnavi Ananthanarayanan is an EMBL Australia Group Leader at the Single Molecule Science Collaboration at the School of...
So I welcome everybody. Thank you for joining in for this rendezvous.
Just give me one.
Today we have with us Dr. Vaishnavi Ananthanarayanan.
Yes. Yeah, sorry. There was some problem at my end. Yeah. So she is currently an EMBL Australia Group Leader at the Single Molecule Science Collaboration at School of Medical Sciences at the University of New South Wales in Sydney. She received a dual degree in BE in Computer Sciences and M.Sc in Biological Sciences from BITS Pilani in 2009. And after that, she spent a year at the Microsoft Research Labs India, situated in Bangalore, as a research assistant. She did her Ph.D. in Biophysics in Max Planck Institute of Molecular Cell Biology and Genetics in Germany. And soon after finishing her Ph.D. in 2014, she started her own independent group at the Center for Biosystem Science and Engineering at IISc. She later became an assistant professor at IISc in 2017, and finally she moved to the University of New South Wales in 2020. Her research interests include how protein motors sort and transport cargo inside cells, and the role of these protein motors in organizing subcellular components like the mitochondria, about which she'll be talking a little of today. And it shouldn't really come as a surprise that she is a recipient of many awards and fellowships, the most recent of which was the Indian National Science Academy Medal for Young Scientists in 2020. Dr. Vaishnavi is also an active advocate for women's equality in science. In June 2020, she co-founded Bias Watch India, an initiative to combat gender-biased conferences, meetings, and talks. So thanks a lot, Dr. Vaishnavi, for accepting our invitation. I think we can start the talk now.
Thank you, Sanskriti, for the very nice introduction. And thanks everybody for making it out here on a Sunday afternoon. I appreciate it. So before I start sharing my screen, I'd just like to tell you that please feel free to stop me at any point of time that you think my talk is unclear. I'd be more than happy to clarify your questions before we move on. So let me share my screen now. Does everybody see this? And my cursor?
Yes,
yeah.
It's with.
Okay, great. So yes, so the title of my talk, as you can see here, is How to Get Away with Fission, the Tale of Mitochondrial Division and Partitioning. So before I move on, I'd like to give you a very quick overview of the questions that we like to address in the lab, all of which are focused on a central theme of cellular organization. We go all the way from single molecules to whole organelles, and we've done a few things along the way. But today, what we're going to talk about is this particular aspect of interaction between something called the cytoskeleton inside cells and organelles such as mitochondria, which form compartments inside living cells. So the first player that I'd like to introduce to you today are these structures called microtubules. So microtubules are the dynamic self-organizing polymer. So this is essentially self-encompassing. This title defines what microtubules are. In living cells, you can do something called staining for microtubules. So you have an antibody that binds to the microtubule protein, the protein that makes up the microtubule. And these can be fluorescently labeled. So when you shine a light a specific wavelength on these cells, they fluoresce, and the microtubule structures show up because the antibodies which are targeted at the microtubules are fluorescent. And when you look at these cells using a light microscope, you'll see something that looks like this. In the same fashion, the nucleus of the cell has been labeled. So you have one single cell here, another here, one more here, 3, 4, and 5. So essentially there are 5 cells here, all of which have been labeled for their nuclei and their microtubule structures. So microtubules essentially are polymers that can assemble and disassemble in living cells. And when you look at what this means, these microtubule structures actually are made up of, are hollow cylinders made up of these dimers of alpha and beta tubulin. It's not super important for you to remember any of these names. It suffices to, it's sufficient for you to know that these are indeed polymers made up of subunits that are heterodimers. And essentially they require a molecule of GTP for polymerization. There's a hydrolysis of GTP that occurs to force them to undergo something called depolymerization. So essentially you can build these structures inside living cells by making use of this GTP hydrolysis phenomenon. And this happens routinely inside living cells to these microtubule structures. So when you look at a living cell and microtubules inside them, again, these are microtubules that are fluorescently labeled. So you can do a simple genetic trick to make the microtubules alone fluoresce inside living cells. And that's what we've done here. And each of these is a microtubule. These tube-like structures is the microtubule. And you can see that they're undergoing these rapid growth and shrinkage events. Growth and shrinkage are equivalent to polymerization and depolymerization, and I'll use them interchangeably. But essentially what's happening here is that when the polymer is growing, you're adding to the end of it. And when it's depolymerizing, you're removing stuff from the end of it, right? So these are microtubules that are essential structures inside living cells for a variety of reasons. One of them is that these microtubules serve as highways inside the cell on which you can have movement of cargo. So this cargo can be many different kinds of things. So mitochondria, for instance, is a cargo. Other kinds of organelles are also cargo, such as the mitochondria. But you also have stuff that needs to be taken inside the cell at specific points in time that can also constitute cargo for these cells. And microtubules are the highways on which the transport of these cargo is mediated by something called a motor protein. So a motor protein is essentially a protein that converts the chemical energy of ATP to mechanical work. And by doing so, it is able to physically translocate on microtubules inside living cells. And this is a movie that I quite like to illustrate this point. So this little thing here is a motor protein that is transporting a cargo. This is something called a vesicle. This is membrane-bound cargo. So essentially this motor protein binds on the one end to the cargo, and on the other end, it takes these steps along the microtubule to bring about movement of large vesicles such as this inside living cells. So in addition to vesicles, you can imagine there's a mitochondrion there, and that also gets transported along microtubules in living cells by motor proteins. So this is, of course, one of the essential functions of microtubules, acting as a highway on which motor proteins. So this is the microtubule here, in case it wasn't very clear. This thing here is the microtubule. This is the motor protein carrying the cargo, right? So this is one of the essential functions. Is there a question? Sorry, I thought I heard something.
There's a question. What's a...
Yeah. So, ma'am, here, how does this motor protein know? How does it stick to this vesicle where it is binding, and how does it deposit? I mean, it has to have some kind of knowledge, that it needs to leave the vesicle at this place, or it needs to attach to it. So how does it recognize? Yeah.
That's a great question, right? So the attachment of the motor to the vesicle in several instances is mediated by another protein that is not illustrated in this simple animation here, but there are proteins called adapter proteins that associate with the motor protein on the one end, and on the other side, they actually are present on the vesicle itself. And the motor protein actually binds to these adapter proteins on the vesicle, right? So that is how the association can happen. Other motor proteins can bind directly to lipids. So I told you these are membrane-bound compartments, right? So that those are made up of phospholipids. So that can be also bound directly by motor proteins in some instances. So the next question that you asked is actually a very, very interesting question. How does a motor protein know when it needs to attach to the cargo and when it needs to detach from the cargo? And that is another question that we've been addressing in the lab. Maybe at some point I'll give another talk and tell you about that. But the important thing is there is no a priori knowledge that the motor protein has that it needs to associate here and dissociate there. So this is a little, although this is a beautiful animation, it's a little misleading in that it seems like the motor protein is always bound to this cargo. That's not the case. There's a dynamic association and dissociation of the motor with the cargo, and that is not illustrated here. So there isn't any a priori information that the motor protein has that it needs to attach here and detach somewhere else. But what does happen is that these attachments and detachments happen stochastically. And that is a beautiful thing, right? So you can ask the question, when something is binding and unbinding from something else stochastically, but needs to transport it from one location to the other in the cell, how do you actually bring that about, right? So that is an amazing question. I'm glad you picked up on that. And that's another question that we've been addressing in the lab using single molecule tools, but that's not going to be the focus of our talk here today. But yeah, like I said, it's a stochastic process, and so there isn't determinism in the way that things happen inside cells, but you do have integration of several of these stochastic events to bring about complexity in organization. And that's a great question. Anything else?
No,
I have a question. Right. So
Pratyush, go for it. Yeah.
So these are membrane-bound vesicles and particles that bind to the cytoskeleton. How do other things that do not have a membrane in the cell move?
So the stuff, typically if things are small enough, you can get around inside the cell by diffusion, right? So diffusion is possible for something that's small. But for something as large as this, you do need motor proteins to effectively transport. You still have diffusion. Of course, the time scales would be much longer if you relied solely on diffusion for something that is this big. So what the cell does is, for things that are membrane bound, which are typically larger, you rely on active transport, which active meaning in this instance, you consume energy to bring about movement rather than passive movement, which is diffusion, right? There's another thing as well that happens. mRNA, for instance, is also transported by motor proteins. In some instances, you actually have hijacking of things that are not membrane bound on membrane bound compartments to bring about their movement as well. So there's actually not hijacking, hitchhiking of other particles. Yeah.
Yeah. So the microtubules in the cytoskeleton visible in the last slide, the red thread-like structures are those, right?
Yes.
The previous
All
slide.
of these thread-like structures are microtubules. That's right.
So why is there this central black void region? What is that? Does that represent?
Oh, no. So each of these is a different cell. So that's just an area on the plate where there was no cells. So, so if this is one cell, this is another. That's just a place where there's no cells.
Right.
Okay. Shall I proceed?
There's an online question on YouTube, I suppose.
Okay.
So, by Kanishka Prarashar. And he's asking, after detachment of one cargo, is there something like another cargo that comes and binds, or detachment of cargo is the end point for motor mobility?
Actually, that's a great question. Okay, without going into too much detail, all I can tell you here is from our research, what we see is that this cargo actually remains close to the microtubule. So the motor, so the cargo is located close to the microtubule. The motor protein binds to the cargo and the microtubule, moves it for a bit. The motor then detaches, leaving the cargo where it is. Another motor can then bind to the same cargo, bring about movement. Essentially, it's almost like a relay race, just that the cargoes are switching out, the motors are switching out, leaving the cargo on the microtubule, essentially. Yeah? Okay, I'll move on here because that's something I don't want to talk about in this talk. This is all about the mitochondria, and we haven't gotten to the mitochondria yet. Yeah? Yeah. Okay. So in addition to serving as highways on which vesicles, other cargo can move on the microtubule, the microtubules perform an essential function of forming a structure called the mitotic spindle. So during cell division, you need to have a proper arrangement of microtubules and their dynamics, in fact, to bring about division of the nuclear material at the time of cell division. So this is, in red, you have genetic material. Essentially, histone is what is labeled here in red. And in green, you have the microtubules. So you see that these microtubules form these thread-like structures that interdigitate with the and attach to, in fact, to the nuclear material. And with specific cues, what will happen is that these microtubules pull apart the nuclear material, and that is how you have effectively cell division happening in these cells. So this is, of course, also involving other motor proteins, but an important component is the microtubule and its dynamics of polymerization and depolymerization. As you can imagine here, there is a period in which it's elongating and then a period in which it is rapidly depolymerizing as well. So these dynamics of microtubules and microtubules themselves are essential during this process. And that is essentially the primary component of the mitotic spindle as well, which is a structure that is responsible for cell division. So this is essentially the function of primary functions of microtubules in cells. And the second part of the equation that we will cover today is mitochondria themselves. So all of you have probably heard of the mitochondria, and I don't want to use the same epithet that people use with the mitochondria because it's done and dusted. I don't want to say what it is. It is not just something that produces energy, but what I want to focus on today is also actually with the way it looks. As you can see here, mitochondria, unlike what you see in textbooks, are not just simple bean-like structures that are just sitting there, but rather they form these large networks of structures that can have a variety of different shapes. So the nucleus, this is a single cell. The mitochondria are in magenta here. The nucleus is sitting right here. And you'll see that the mitochondria form these structures that are very different from what you have probably seen in your textbooks. Some are very long, some are short, some are circular. So you have all sorts of shapes and sizes. So this is, of course, something which is interesting. What's even more interesting is the way that these mitochondria move inside cells. So this is looking at just the mitochondria in some cells. And what you'd see, this is actually from a recent video that I came across on Twitter from Andy Moore. And what you can see is these really nice movements of mitochondria. They're wiggly, they change shapes, they do all sorts of things. And essentially, what I want to impress upon the audience here today is that the mitochondrial network in the cell is extremely dynamic. It undergoes the mitochondria, each mitochondrion is capable inherently of undergoing fission as well as fusion. So fission is essentially splitting above one mitochondria into two. And fusion is, of course, coming together of two independent mitochondria to form one continuous network. So this mitochondrial fission and fusion is actually maintained in a delicate balance in most living cells. And if you perturb this balance even very slightly, you end up having something called mitochondrial dysfunction. Essentially, dysfunction is improper functioning of the mitochondria. So the shape of mitochondria relates to its function. And if you have an imbalance in the fission and fusion of mitochondria, you end up having mitochondrial dysfunction. Another important thing to remember here is that mitochondria need to be passed down from one generation to the other during cell division. So in the movie that I showed you earlier, where microtubules were pulling apart nuclear material, it was also essential for the mitochondria during that step to have been divided semi-equally, at least, between the two resulting cells. And that is because mitochondria cannot be produced out of nothing or de novo in the cells that have just been formed as a result of cell division. And this is, of course, to do with also the fact that mitochondria contain their own DNA. Some of the genes that are transcribed in the mitochondria actually make up proteins that form essential structures in the electron transport chain where the energy production takes place. So structurally and functionally, there are proteins that are being expressed that reside on the mitochondrial DNA. And so if you don't have mitochondrial DNA from the previous generation, you will not actually form these proteins that make up the essential structure of the mitochondrion. So therefore, there needs to be a mechanism wherein you ensure that the mitochondria do indeed get separated from the original parent into the two cells that are formed as a result of cell division. So essentially, mitochondria are super dynamic, and they also need to undergo division at the time of cell division, so that you ensure that the progeny actually have mitochondria as well. Any other questions until now?
Yeah, there are a few questions.
Yeah.
So, Vatsal, would you like to unmute and ask?
Yeah. So, how many mitochondria are typically in a cell, or in the cell that you show? That
I will show you statistics for the cells that we worked with, but the short answer is it's variable. Even for the same cell from one cell cycle to the other, there are changes in the number. What's important is also the volume of mitochondria, because you can have a total volume that is invariant, but you can have different numbers of mitochondria depending on what the form of the mitochondria is. For instance, you can have one long mitochondrion or a few shorter mitochondria. The numbers are different, but then the total volume could still be the same, right? But that's an interesting question, and I will talk about that as we go along.
right, right. And one more question. So you said mitochondria are not produced de novo. So are there other organelles which are produced de novo, like ribosomes or Golgi apparatus?
No. So ER, for instance, can be. So the primary reason why that can't be done for mitochondria is simply because of the fact that the proteins that make up, for instance, the inner the mitochondria has two membranes, right? Outer mitochondrial membrane, inner mitochondrial membrane. A lot of the inner mitochondrial membrane proteins are participating in electron transport, and those proteins are produced by the genes that are present within the mitochondrial DNA. So mitochondrial DNA, of course, within the mitochondria themselves. So if you don't have these being passed on, you cannot actually make mitochondrial inner membrane, because the proteins that make up at least the electron transport chain, there are about 30 something genes that are actually required for mitochondrial functioning, as well as the structure that are residing in the mitochondrial DNA. That's why you can't have them being produced de novo. ER, for instance, you can do that. Golgi also, I think you should be able to produce them de novo. But for mitochondria, you cannot do away with it.
Right, right.
So Chandrasekhar also has a question, but he's asked me to speak it from his side. So he says, fission is making sense to him, but why would two mitochondria want to fuse together?
I will tell you in just another slide.
Okay. Avni, do you want to unmute and ask your question?
Ma'am, I wanted to know what kind of perturbations exactly cause the dysfunction.
I will show you, if you can hold on.
Okay.
Okay, that's it.
Is that it? Okay, cool. So yes, there were questions as to what happens, why do you need mitochondrial fission and fusion, right? So the form of the mitochondrion is inherently linked to its function. These are, of course, so the way that these studies have been done varies. So you have different model organisms in which these experiments are done. You have different conditions in which the experiments are done. But there is some consensus that that is emerging that suggests that mitochondria that are more fused are better able to produce energy, ATP essentially, as well as you can imagine an instance where you have mitochondrial damage, either DNA damage or essentially mitochondrial DNA damage that leads to dysfunction in the mitochondrial production. You could dilute out the mitochondrial damage by allowing it to fuse with a healthy mitochondrion. So these are two ways in which you can imagine mitochondrial fusion being important. So essentially mitochondrial fusion increases the ability of the mitochondrion to effectively produce more energy. There's not just that, you also have calcium homeostasis and other functions of mitochondria that are usually overlooked, but all of these are much better controlled when you have longer mitochondrial networks. On the other hand, mitochondrial fission is not just required to be able to ensure that there is equal partitioning of mitochondria, which is something I'll show you later, but you also need mitochondrial fission to be able to effectively control the damage that you have in the mitochondria. So for instance, if this mitochondrion, so imagine if this mitochondrial bit on the right is damaged. What you could then perhaps do is only take out that part away and make it go to the lysosome for degradation. So essentially what happens is a process of mitophagy, which is essentially the cells eating of its own damaged mitochondria, that kicks in to make sure that mitochondrial damage is kept at a minimum. Because what happens with mitochondrial damage is that you have production of something called excess production of reactive oxygen species, which can be deleterious to a cell. So what happens as a result here is if you're able to effectively take away just a portion of the mitochondrion that is damaged, that will help you prevent excessive damage from reactive oxygen species. So this is essentially a way of mitochondrial quality control that makes sure that fission and fusion make sure that mitochondrial quality control is maintained. And that is one of the primary reasons why you do have both of these happening. That's not to say that let's say if you take away the mitochondrial fission proteins and the mitochondrial fusion proteins, mitochondria will still be fine until there are stressors that they come across. And in which case they will start acting up and there'll be a problem for the cell eventually. Yeah, so I hope that answers the questions that others had previously. Right. Just to quickly also take you through what mitochondrial fission and fusion proteins do to the mitochondria. Don't have to worry about these names here. You can forget all of them. All I want you to take away from this slide here is that mitochondrial fission results in the formation of 2 mitochondria from 1, 2 of course, which are smaller than the one that was initially there. And mitochondrial fusion, of course, results in the formation of a larger mitochondria. And there are different sets of proteins that mediate these fission and fusion reactions. So this is a cell in which the mitochondria are labeled. You probably can't see it super well, but then there is again a distribution of mitochondrial sizes, as we saw in the example cell I showed you previously. If you take away the mitochondrial fission machinery, which is what has been done to the cell, you'll start seeing that there are these long networks of mitochondria. So essentially what you're having is, instead of a distribution of mitochondrial sizes, you just have long networks of mitochondria. And conversely, if you take away the mitochondrial fusion machinery, so again, you're looking at normal cells where you have mitochondria that have a distribution of sizes. When you take away the mitochondrial fusion machinery, you'll start seeing these almost punctate mitochondria form because there is hyperfission of mitochondria. So the balance between fission and fusion is what's being affected here. So in this instance, when you took away the fission machinery, there was excessive fusion. And conversely, when you took away the fusion machinery here, there was excessive fission, resulting in these fragmented mitochondria. And in addition to these fission-fusion machinery, like I mentioned to you previously, mitochondria travel along microtubules, mediated by motor proteins, of course. This is a portion of a neuron, an axon, in fact, where you're looking at just the mitochondria that are fluorescently labeled. And what you can probably see here are these really interesting looking train-like things that are moving in both directions. These are in fact just mitochondria moving along the microtubules in both directions. There are motor proteins that mediate movement in both directions. And that's what these are. So essentially, these are mitochondria being transported by motor proteins along the axons on microtubules. So these are the range of dynamics that you can have the mitochondria perform that is essential for their function. So now I'm going to actually move on to telling you a little bit about the same proteins in yeast. The same proteins exist in yeast, and I'm going to tell you that we ask some of the questions we do in yeast because of our ability to actually be able to visualize them very nicely. But suffice to say at this point that the same kind of proteins that mediate fusion and fission are present in yeast as well. This is the example from budding yeast. This is regular cell with the mitochondria that have a range of morphologies. If you take away the mitochondrial fission protein, you have one long mitochondria that has these really funky looking structures. If you take away the fusion machinery, you now have a lot of punctate mitochondria. So this is with regards to the fission fusion dynamics. With regards to the separation of mitochondria at the time of cell division, it's known how this happens in budding yeast. I'll show another video here. Very nicely, people have shown how some other cytoskeletal structures are required for the movement of mitochondria to the newly forming bud, which is how these cells undergo division. But in fission yeast, this undergoes symmetric division, much like bacterial fission. It undergoes separation in the center, which is why it's called fission yeast. This is, of course, asymmetric cell division because you can see that one of the cells that are being formed as a result of cell division is smaller. And in this instance, you have motor proteins moving. Sorry, I'll go back there. This instance, you have motor proteins moving mitochondria into the newly forming bud. But here in fission yeast, there are no known motor proteins that actually move mitochondria to either cell end to mediate the partitioning of mitochondria. So this term for making sure that the mitochondria are divided and distributed between the two cells formed from cell division is called partitioning. So there is no known motor protein that mediates the partitioning of mitochondria in fission yeast. But what's known is that it is a microtubule mediated process. It's not entirely clear how the microtubules are involved, but all that's really known is that the microtubules are somehow involved in this process. So this is one thing that happens in fission yeast, but the question we really wanted to address here is how does this mechanism of microtubule mediated mitochondrial positioning and partitioning occur inside living cells? I say positioning here because that is also something that has been looked at in fission yeast. It's known that the mitochondrial positioning inside cells, the way it appears inside a living cell, also relies on microtubules. So this is the question we set out to ask. How does this microtubule mediate mitochondrial positioning and partitioning inside living cells, living fission yeast cells?
Yeah,
I will introduce, so I'll just quickly introduce fission yeast, and then if there are any other questions, I'll take them then. Is that all right?
Yeah, sure.
Because I just wanted to show these images, just this one slide. So essentially fission yeast are these cylindrical structures that undergo division right in the center. This is the mitochondria inside fission yeast cells. And as you can see, they also have these nice range of distributions of sizes. And they also are very few in number. So you can actually count each mitochondrion very nicely. These are microtubules inside fission yeast cells. And you can see that they also form these structures that are long and similar to what we saw in mammalian cells, except of course, there are very few here. Again, we can easily take one microtubule, track it for its duration that it's visible, and you can still do very well with just going from one cell division cycle to the other even. So it's a very nice model organism to work with to understand microtubule mediated mitochondrial positioning. And we use this as the model organism to understand how this happens in other mammalian cells, such as our own cells, because our own cells also rely on microtubules for positioning, as well as for partitioning of the mitochondria. So this is why we use fission yeast. This is also a eukaryote, just a single cell eukaryote. And it also relies on the microtubule and the cytoskeleton for mediating the positioning and partitioning of the mitochondria. So for this work, we use this fission yeast as a simple model organism to understand how this process happens. Yeah, any questions now?
Yeah, so I have this confusion that when we are saying microtubules are mediating the partition, so are individual mitochondria being fissioned into two, or during cell division, half of the mitochondria are being pulled to one cell and half to the other?
I'll show you. I'll show you what it is that happens. That's the crux of the talk. We're going to understand how that happens. Are there any other questions?
No, I don't see any.
Okay, great. So I'll move on. So this is the basic premise. I'm hoping everybody is here still with me. If something is unclear, do let me know and I'll try my best to explain it to you. Yeah. Okay, so this is the question. How are we going to address it? So this is actually a rendition, 3D rendition of a fission yeast cell in which the nucleus is in purple. So this is actually from this cryo-EM tomogram. So essentially what you're doing is at low temperature, you're cutting through a fission yeast cells in very, very thin slices and looking at it with EM technology, electron microscopy, basically, and then putting together these slices in 3D so that you have a rendition of structures inside the cell. So when you do that, you can see the entire cell with very high resolution. So that is, of course, the primary difference between electron microscopy and light microscopy. You use visible light during light microscopy, and electrons in electron microscopy as an illumination source. And because of the wavelengths that you use, you have much higher resolution with electron microscopy. And so you can actually see very nicely all the structures inside living cells. So you have the nucleus here, and then you have these green thread-like structures that are actually the microtubules. The blue blobs are the mitochondria. So what you can probably clearly appreciate here is that the mitochondria are laying very close to the microtubules, right? So this, in fact, has been gone on to be studied. And what others have determined is that there is an actual protein that links the microtubules with the mitochondria in fission yeast. So essentially, the protein is called MMB1. We won't need this until much later. I will refer to it as a linker protein just so it's clearer. But essentially, this linker protein links the mitochondria with the microtubules. And without the linker protein, you can actually do genetic knockout of the linker protein. And what happens then is that the mitochondria no longer attached to the microtubule, right? So this is primarily what happens in fission yeast. The primary way that microtubules interact with the mitochondria is via this protein, the linker protein. So I showed earlier a video in which inside a neuron, where you were looking at the movement of mitochondria on microtubules mediated by the motor proteins that we discussed earlier. So this kind of movement of mitochondria on microtubules does not occur in fission yeast. And one of the reasons may have to do with the fact that the cell is in fact rather small. When you compare it with a neuron, which can be up to millimeter in length, the fission yeast cell is only about 7 microns in length along the long axis. And that is not a very long distance. And there isn't any microtubule mediated, motor protein mediated movement of the mitochondria on microtubules in fission yeast. So there is a slight difference, of course, in between these two eukaryotic cells, but still there is an involvement of microtubules in mitochondrial positioning in these cells. So we'll go ahead and see how this looks like. So what we did was to actually employ these cells in which you can again see the microtubules and the mitochondria. So the microtubules are in green. So these are individual fission yeast cells. So there are about a few tens of cells here. And what you can see in green is the microtubules. Again, they look not unlike what you see here. You have these microtubule structures. Mitochondria are primarily along the microtubule network. There are, of course, deviations in which there aren't attachments between microtubules and mitochondria, but primarily the mitochondria seem to positioning seems to be dictated by the microtubules. So one of the first things we did was to see what happens if the microtubules are taken away. And you can do that by adding a drug which inhibits microtubule polymerization. So what that means is that the microtubules will be completely depolymerized. So this is just a stub of the microtubule that you see here. The white dots here are the remnants of the microtubules that were present previously. They used to look like this, these long thread-like structures. They're now gone because we've added the drug that depolymerizes microtubules. There's only a remnant of the microtubule. And when we do that and looked at the mitochondria, you start seeing that the mitochondria are actually fragmenting quite a bit. What looked like this is now looking like this. You can see that there are dots that are actually moving now. They're diffusing a lot more, of course, because they're much smaller. But what in this cell, for instance, you can start seeing that in the first frame, they were quite long and then they start fragmenting, right? So what we can immediately see happening here is that mitochondria undergo fragmentation if you depolymerize microtubules. So this was interesting. And this is a nice observation that we've had. We wanted to explore further what was actually the interaction between the microtubules and mitochondria. So using the drug is, of course, very nice, but then that is like a brute force approach wherein you're taking away the microtubules altogether. What we wanted to do, is that a question?
Yeah, there are a few questions actually. There are 5.
Okay.
So, yeah. Let me start with so there are a few questions on YouTube that I'll start with. So Kanishka is asking, as per the endosymbiotic hypothesis for origin of mitochondria, is the mechanism of fusion or fission an evolved mechanism, or it can be supposed to found earlier too?
No, I don't know about how the endosymbiotic theory fits into the fission fusion. Fission, of course, makes sense because you do have fission of bacterial fission, which could make sense in context of just the division that bacteria undergo. But fusion, I'm not sure if there is an explanation. I wouldn't know that. Sorry. Yeah.
So she also is asking, what is the specific localization, cellular localization of mitochondria? Is it always near the nucleus? Typically not. It's usually distributed along the entire surface of the cell.
There is, of course, a propensity for some of the mitochondria to appear around the around the nucleus in eukaryotic cells. And that depends on the way that the motor proteins are interacting with each other to mediate movement along the microtubules. But in this, for instance, in fission yeast, as you can see from here, sorry, they are along the entire length of the microtubule. So this is what you'd expect, because you do not, especially in a cell like the neuron, you actually have ways in which you can make sure that mitochondria are positioned at regular intervals along the entire length of the cell, because there could be instances where you have lacunae in energy production if you don't actually distribute the mitochondria, especially in a cell like a neuron.
So I have a question about this particular image. So this somehow looks, it doesn't look very, so you said this has been created by sections of electron microscopy, right?
So yeah, so you image sections using electron microscopy. This is a rendition from the electron microscopy image. This is not, of course, what you would get as a raw data from electron microscopy. This has been colored and 3D, yeah.
So the machine allows you to do all of that. The electron microscope.
Sorry?
The electron microscope software probably allows you to do all of that.
Yeah. So once you acquire the images, you can stitch them together. There's a bunch of different kinds of software that you could use. These are not our images. This is important. No, this is from a different group from a while ago, in fact. Yeah.
Okay. So the question by Raj Rishi. Raj Rishi, you want to unmute and ask?
Hello. Yes, ma'am. Ma'am, so as you were talking about the fusion process of the mitochondria, so ma'am, what happens to the complexes that are present outside? For example, the Tom complex and the Sam complex that we have seen. So what happens to those specifically?
So there are different machinery that are required for inner mitochondrial membrane fusion and outer mitochondrial membrane fusion. And so essentially these are membrane complexes, right? So they also form one continuous membrane at the end. So essentially, if I go back to the, so essentially this is just showing these proteins that are involved in the fission and fusion. But if you look at OPA1, that is required for inner mitochondrial membrane fusion. So presumably what happens when two independent mitochondria fuse is for it to form one continuous membrane now. So essentially the proteins that are found on the membranes of one of these mitochondria will be continuous with that of the other now. And that's because the fusion of the membrane will essentially make sure that the continuity is maintained. That is the inner mitochondrial membrane, of course. And the outer mitochondrial membrane has another set of proteins, and that is also, so MFN1 and 2 are the outer mitochondrial membrane fusion proteins. So this is, of course, a complicated process, which, although it is relatively simplistically shown here, fusion and fission, for that matter, are quite energetically involved processes. So all of these proteins are in fact GTPases. So they require GTP for the fission and the fusion. So once the fusion and the fission processes are complete, you would end up with distinct compartments that have a continuous membrane, which have mixtures of proteins from, when in the case of fusion, mixtures of proteins from both of the mitochondria that form the original two parent compartments. In the case of fission, of course, you're going to be separating one compartment into two. So you'd have mixing, essentially. Short answer for a very long explanation is that you'd have mixing.
Ma'am, so the single mitochondria would be more beneficial because it would have more signals to the translocation part.
Sorry, can you repeat that? I didn't get that.
Ma'am, so this a single large mitochondria would be more beneficial to the cell, right?
Not necessarily.
It would have.
I wouldn't say that that's the case. So for one of many reasons, a single long mitochondrion wouldn't be beneficial. First of all, because it's harder to move that long mitochondrion. So in a case of a bigger cell, for instance, that would likely be localized to a specific region. And that is less than ideal, especially in the case of high energy demanding cells like your neurons or your muscle cells, where you do need effective distribution of ATP along the entire length of the cell. So that will be an issue. The other thing is, of course, when it comes to dividing cells, you'd actually not prefer to have a single long mitochondrion. I'll show you later why that is the case, based on what we see and also what's known in the field.
Thank you.
Sure.
Yeah, I have a question. So, how many proteins that you talked about are involved in the electron transport? So, how many of them are actually present in a typical inner mitochondrial membrane?
I'd say hundreds. I don't know if anybody's actually counted it, but why were you asking this?
So this is because when you said that suppose there's one part that is damaged and then that part of the mitochondria is fragmented away and degraded by lysosome. I was thinking what part of how much of the electron transport machinery would be degraded, and how much the efficient, how much the output in the terms of ATP production would be lowered in those cases.
So there is a trade-off that you'd have between so there's not going to be effective energy production if you stick with the damaged mitochondria as well, because you also start producing reactive oxygen species which damage the mitochondria and the cell. So of course that's a trade-off between how much energy you can produce, but given the fact that you have damaged mitochondria, it's probably going to be deleterious for you to keep the mitochondrial structure intact without getting rid of the damage. So overall, I think it would be difficult for a mitochondrion to function if it did have mitochondrial DNA damage in any case. So it wouldn't make sense for it to retain its structure with the damaged mitochondria still within it.
Yeah.
Okay, so I will go to the part of the talk I was at. Yeah. Oh, okay.
At the end.
Yeah. Okay. What's the time?
It's 12:52.
Oh gosh, I've already been speaking for 50 minutes. Okay, I will be very quick then. Okay. All right. So basically, the question we asked is, we can make microtubules longer and shorter genetically. So what happens to the mitochondria in this instance, right? So these are cells in which the mitochondria are like regular wild type cells. These are cells where the mitochondria are long. These are cells where the mitochondria are short. Microtubules, sorry. Microtubules are long and microtubules are short. You can just remember them with a shorthand of microtubule long and microtubule short. There's no necessity to remember the names. But essentially, what we found was if you make microtubules longer than normal, you actually make mitochondria longer than normal. This is regular cells where you have effectively a range of mitochondrial size distributions. If you make microtubules shorter, you make mitochondria also much shorter. So this is 3D renditions of the mitochondria and microtubules in these cells. And the interesting thing is, like we saw before, the number of mitochondria per cell changes. In typical wild type cells, you only have 5 mitochondria, which becomes only about 2 or 3 in cells with long microtubules. And in cells with short microtubules, you actually have many more mitochondria. But like somebody was asking me previously, what really matters is the mitochondrial volume. Because if you look at the total mitochondrial volume in these cells, you actually do not see a difference, indicating that this change in mitochondrial numbers arises not from a difference in how much mitochondria it produces. You can have a process of biogenesis of mitochondria where you put together different components to make up a mitochondrion. So that is not changing between these 3 mutants. What's actually changing is just the way that the mitochondria are presented, the form of the mitochondria essentially. And that is important. So essentially by changing microtubules length, we're changing mitochondrial length and therefore number. So the next question we asked is, what is the basis for the change in the mitochondrial number? And this is a whole field of Fission cells. So you can pick any of your favorite cells. Ignore the really bright ones. They've just taken up too much of the dye. But if you look at any other cell, you'll see that there are mitochondria within them and that they are doing these strange wiggly things. But if you can see in this one particular cell, for instance, there was a fission event that happened just over there towards the end of the movie. There you go. So you can look at fission and fusion of mitochondria in these cells. And what we did was to actually track fission and fusion in these cells. So you can actually see them undergo really nice fission and fusion. And we saw how this happened. I will skip this slide, but what I will tell you is we saw that the mitochondrial fission frequency and fusion frequency was different between the three cases. So the mitochondrial fission frequency was something, or fission and fusion frequency was something we measured to ask if the changes in mitochondrial numbers were because of a difference in fission or fusion, right? So you can imagine that if you had increased number of mitochondria, you could have increased fission or reduced fusion. So what we wanted to ask here was how is it that the change in mitochondrial numbers are happening? So these were, of course, different fission and fusion values. For the wild type, there was some number, but for the long cells with long microtubules, there was a reduced fission. Cells with short microtubules, there was increased fission. And this was largely replicated in case of fusion as well. But remember, these are mutants cells, which means they are stable and at steady state. So you would expect that the fission and fusion frequencies are in balance with each other to maintain the steady state of number of mitochondria. So to actually understand what is happening as a result of microtubule loss, we went back to the first experiment we did, which was to depolymerize all the microtubules. So basically taking away all the microtubules and asking within a short period of having taken the microtubules away, what happens to the mitochondria? And that's what you're looking at here. These are wild type cells where we've added the microtubule depolymerizing drug. And these are cells with long microtubules where we were adding the drug. And at the end, you can start seeing these nice fission events. When you look at the number of mitochondria over time in cells where there's been no drug added, wild type cells where there's no drug added, of course, you'll see that the mitochondrial numbers are remaining fairly stable. If you now add the drug for depolymerizing microtubules to these cells, you start seeing a nice increase over time. Same thing in the case where you have long microtubules. So you have fewer mitochondria to begin with, the orange line that is, but then that also picks up. And at the end, you have many, many more mitochondria. Now, the interesting thing here is, although there was an increase in mitochondrial fission frequency, as was visible here as well, of course, there was no change in fusion frequency. So this is confirming our earlier hypothesis that the change in mitochondrial numbers, the increase in mitochondrial numbers upon changing the microtubule length, in this case, total loss of microtubules, results from increase in mitochondrial fission frequency. So this is essentially an important point because what is happening as a result of loss of mitochondrial association with the microtubule. So essentially, mitochondria are bound to microtubules in these cells. You take away the microtubules, you take away the association as a result, and so you start seeing increased fission. And this is exactly what's happening in the case where you have short microtubules as well. The short microtubules no longer effectively interact with the mitochondria. There's fewer attachment points, and so there's increased fission of mitochondria. And this is an important point because there is a biological role for this attachment of mitochondria to prevent increased fission. And that has to do with partitioning of mitochondria at the time of cell division. So in fission yeast, at the time of cell division, you have microtubules that are arranged like this. And it forms something called the mitotic spindle that we saw earlier within the nucleus. So you might have learned that in other cell types, the nuclear envelope first breaks down when you have mitosis starting. That does not happen in fission yeast. You actually have an intact nuclear envelope. And the microtubules go into the nucleus to form the mitotic spindle. And what happens here is what we looked at to the mitochondria, of course. So this actually replicates a system wherein the mitochondria, which are going to be within the cytoplasm still, of course, they can't go into the nucleus, are left free of microtubules. So what we'd expect is that there is increased mitochondrial fission. And that is exactly what happens. There is increased mitochondrial fission when the mitochondria are when the cell goes through to mitosis. And in fact, if you look at a time lapse of this process happen, we can actually catch events of mitochondrial fission. And the question we asked is this increase in copy number of mitochondria sufficient to separate it between the two cells during cell division? So here what we're asking is, can a single mitochondrion do something like a coin toss to determine whether it goes into one daughter cell or the other? And essentially, you can do that by counting the number of mitochondria in the mother and the daughter and compare it with a binomial distribution with a probability of 0.5. And when you do that and look at the expected, looking at the difference between the observed versus the expected values, we can actually say that they're not different. So indicating that there is a stochastic partitioning of mitochondria at the time of cell division. And this stochastic partitioning works in this instance because you do need increased numbers of mitochondria for this to have low error rates. I'm rushing through this a bit, but if there are more questions, I'm happy to take them in a bit. I'll finish off in maybe another couple of minutes. But essentially, you can imagine when you have only 2 mitochondria, there is let's say you have a single mitochondrion. There is 100% probability of error because only one of the cells that will, 2 of the 2 cells that are formed at the time of cell division, only one of them will get one of the mitochondria, right? So there's 100% efficiency, which is what partitioning error, which is what you see here. As you increase the number of mitochondria, you'll see that the partitioning error reduces. So this is essentially an important point, that you need to have increased fragmentation of mitochondria before you have cell division so that you reduce the error in partitioning of the mitochondria. I'm going to skip this part.
So there are actually a lot of questions. People are.
I know, I'm finishing my talk right now because of that. I'm going to.
You can take your time if you're not in a hurry.
No, I'm happy to. So this is anyway, this is just confirming that there is increase in fission of mitochondria. This is just the mechanism by which it's happening. This is not super important. So I'm going to skip through this and show you the final conclusion here, which is essentially all I have told you right now, which is that when the mitochondria are bound to microtubules, fission is inhibited of the mitochondria. When the mitochondria are released from the microtubule. So this happens because, as I told you, in living cells, microtubules undergo polymerization and depolymerization. So when this mitochondrion that is bound to the microtubule, the underlying microtubule undergoes depolymerization, this mitochondrion is now free. And so what happens is that it undergoes increased fission. And this increased fission of mitochondria, when the microtubules are not present, is essential for the way that the mitochondria are partitioned in these cells, which is by a stochastic segregation mechanism, right? So essentially, this is the whole story. In future, what we want to understand is how this might be playing out in context of neurodegeneration. What we have here is essentially a system where we can relate the microtubule dynamics and say that that is what causes fragmentation of mitochondria. Interestingly, in neurons that are undergoing neurodegeneration, what's been studied is that the microtubules are actually not so great at remaining a specific length, meaning they are not stable because of production of proteins that happen during Alzheimer's and Parkinson's. So you have fragmented microtubules here. Also, what's known is that mitochondria undergo fragmentation in these cells. So in this context, I hope you can't hear my cat, he's going a bit crazy. But in this context, what we're trying to ask is if we can understand how these two might be related based on the clues that we've got from fission yeast. So this is where the neurodegeneration angle comes from. We're going to be exploring this in the future. So I'll stop here and take all the questions that you have. Sorry, it went a bit over, but I think, yeah, I'm hoping people at least got a bit of what I said today. I'm happy to take questions now. Yeah, go for it.
Yeah. So I would like to thank you first for the talk.
That's okay. Yeah.
So there are a few questions. So first off, in the very first experiment that you talked about, this relation between microtubules and mitochondria.
Mm-hmm.
So how exactly is the microtubule so controlling mitochondrial division? That's so
exactly. So this was the part I had to skip, unfortunately. So basically, I told you about microtubules undergoing polymerization and depolymerization and living cells, right? Even without any external cues, on their own, microtubules undergo this repeated cycles of polymerization and depolymerization. For instance, in this one instance, this microtubule is extending until here. It could start depolymerizing and going all the way to this point here where it starts from. And in that instance, this mitochondrion is going to be left free, right? Essentially in the cytoplasm. So what we went on to show was that when this mitochondrion is left free, that is when the fission protein is able to attach to it and bring about fission. That is how this happens.
Okay. So your experiment actually verified this.
Yes, when the mitochondrion is attached to the microtubule, we see that the DNM1 protein, which is a mitochondrial fission protein, is not able to bind to the mitochondrion.
Okay.
So I have a related question. Just what we see in fission, that when it is attached to microtubule, fission does not happen. So can fusion happen in those cases?
We don't think there's a reason for it to not happen. We haven't explicitly checked for it, but we don't see a difference in the fusion rates when the microtubules are present or absent. So given that is the case, we do think that there is no issue with the fusion. Only thing, of course, fusion is rate limited by the fact that you need to have two entities come together. That is what the primary factor is for fusion.
Right.
Yeah. Hi, I have actually a question with respect to this, how microtubules decide over the mitochondrial fission. I just wanted to know, is there any promise with regards to this study? I mean, what would be the implications of this study if mitochondria, as if your results show, decide somehow the mitochondrial dynamics, what does it implicate? Or would there be any significant thing?
That's right.
In
so
terms of
that's exactly what I was leading to, right? So we're exploring this. Of course, fission means we can't study processes that cause disease. Of course, that doesn't make a lot of sense. So which is why we're exporting some of the stuff that we learned to ask similar questions in relevant contexts, for instance, in neurons and neurodegeneration, because there too, in neurons as well, a majority of the mitochondria are actually attached to microtubules stably. So in the movie that I showed you in the introduction, you'll see that a lot of them are actually about 70 to 80%, in fact, is the number, are stationary and stuck to the microtubule. A very small portion are the ones that are moving on the microtubule. So given that is the case, there is a possibility that a similar mechanism could be taking place in neuronal cells, and that's what we're exploring in the future.
Yeah, but in that case, for example, in neurodegeneration, as you showed that schematic, in that case, if it is happening because of hyperphosphorylated tau or something like those mechanisms, those reasons, and at the same
Tau
time,
is
how
a
would
microtubule
you?
associated protein. That's how.
Yeah.
So tau is a microtubule associated protein that is responsible for stability of microtubules.
Yeah.
So when you
what I'm saying.
hyperphosphorylate tau, that destabilizes microtubules, and that's why we think,
yeah, that's the thing that I'm saying. So if it is happening because of the hyperphosphorylated tau or those reasons, how would you establish that the dynamics are getting affected solely because of the microtubular these?
That's what we put in an application for a grant for. That's something I can't divulge, of course, but yeah, that's something we're looking at.
Okay, thank you.
Okay. So there's a question on YouTube. So back then, when you had showed that 3D rendition of the cell, Detach says that the microtubules in that images they seem very sparse.
In which one again? Sorry. Yeah, there are very few microtubules. Yeah, in Fission East, right? In the EM is what they're talking about.
Yeah.
Cryo-EM tomograms. Yeah. The number of microtubules is very, very small in Fission East. So there's about 4 or 5 bundles in these cells. So which is why it makes our lives much easier to be able to see all these microtubules and mitochondria. Yeah.
I have a question. So the more number of mitochondria that we have, better we'll be able to separate them between the cells, right? So you are able to see this phenomena in fission yeast cells because the microtubules are formed inside, right?
Yeah.
There is no nuclear fragmentation. So how is this happening in human cells where nuclear fragmentation will happen? The membrane will be fragmented in.
So that's a great question. So in mammalian cells, what's happening? So I didn't speak too much about this fission protein, but you can control the activity of this fission protein through the cell cycle. In mammalian cells, in fact, what happens is you upregulate the activity of this fission protein by phosphorylating it and so on. But essentially what happens is you have an increased activity of the fission protein during mitosis. And that seemingly is sufficient for you to fragment mitochondria during, that's a great point as well. You brought it up. Even in mammalian cells, you have an increase in copy numbers, effective copy numbers of mitochondria during cell division to partition them. And that happens because you increase the activity of the mitochondrial fission protein and not because of this mechanism. Yeah, absolutely.
Okay, thank you.
Okay, so John, would you like to ask a question?
Yeah, I'll ask. So you had mentioned that the length of the microtubule dictates the length of the mitochondria. So how exactly is the length measured? Like, do you measure from the nucleus to
of the
where it
microtubules,
ends,
the
or is
mitochondria, microtubules.
Microtubules.
Yeah. Yeah, we've done it from, so typically the nuclear envelope contains the microtubule organizing centers. And so that's where the microtubules originate. So we go from the nucleus to the cell end. That is the length of the bundle. The other complication here, which I didn't speak about, is that the fact that these are not independent individual microtubules. In fission yeast, you typically have bundles of microtubules. For all practical purposes, we consider this to be a single microtubule. But yeah, effectively, we measure it from the cell center towards the tip of the microtubules. Yeah.
So when you say it's short, does it not go to each other? Is that the case? When you say it's a short microtubule,
yeah.
When you say it's a short microtubule, does it not go to the edge of the cell?
No. I can show you the images again, just so it's clear. Oh, yeah. Yeah, you see this? So these are some small microtubules that don't extend all the way out.
Okay.
So, yeah.
Yes. So do you not see like mitochondria which are bunched around the nucleus in this case?
So this is what the microtubule. So the mitochondria are free to diffuse here. So typically in these cells, the mitochondria don't diffuse too much because first they're large, and of course a lot of them are anchored to the microtubule. In these, a lot of them are actually not bound to the microtubule, so they actually diffuse out more. So they do end up around the entire cell.
Okay, so
the cell
smaller,
is
like
somewhere
it's more
around
free
here.
to diffuse.
Yeah, it's smaller. Yeah.
Okay.
So like your work says that when you shorten the microtubule, they divide more, right?
Yeah.
So
they have undergone fission. Yeah.
Maybe that is why we can see such small and a large number that is diffused around.
Precisely. Yeah.
Okay.
Also, I have a similar question related to this. In the beginning, you had showed about the mitochondria, which is fusion inhibited and fission inhibited. So even in that case, I noticed that in one of those cases, it's more the mitochondria are more uniformly spread across the cell, and in one case, it's more bunched around the nucleus. So is that related to?
Were you talking about the yeast, this part, or this other one?
Yes. This one, the Mitochondria Dynamics. Yes.
So which one are you talking about? So here I can't tell you where the nucleus is. This is it's not very clear to me where the nucleus is. In here, it's probably somewhere around here. But I wouldn't say they're really bunched up. So for instance, here, this is the nucleus, and these are the mitochondria. So their attachment of mitochondria to the microtubules actually ensures, and the motor proteins, ensures that there is a relatively equal distribution around the entire nucleus. You can perturb this symmetry, of course, by making one kind of motor protein bind to the mitochondria more. And in that instance, you actually do see that the mitochondria are bunched around the nucleus. But I wouldn't call this bunching around the nucleus because, okay, so the other important thing to remember is that you're looking at either a 3D projection, a 2D projection of a 3D object, right? And if you look at, I don't know if I have an image of a cell here, but if you look at a cell section, cross section, the nucleus, it looks like a fried egg, basically, right? So the yolk is the nucleus, and the white would be like the rest of the cytoplasm. So it's very, very thin when it is away from the nucleus. So there's more room for the nucleus, of course, and there's as a result, there's also more stuff that is visible there because of the fact that there's more room. But if you looked at it per volume, it's likely going to be the same amount of mitochondria that you have.
Okay, yes.
If that makes sense.
Yes, I think so. So like in these two images, like the one on the left and one on the right, are they like two different cells? Like two different types of cells?
These are from very different papers, very different cells. I just took them to show how they looked.
Okay.
Yeah. Can you actually go to that slide again? I've been thinking about that thing where you showed a relation of neurodegeneration with, which is your future question.
This is our question. Yes.
Yeah. So I want to understand this question in a little bit more. If you can just explain to me, how do you want to relate these things? Yeah.
That is going to be our future question. I don't think I can give you any more details.
Not in that detail, just the overview.
So we're asking how the microtubule destabilization, if the microtubule destabilization could be contributing to mitochondrial fragmentation. That's the question essentially.
Okay. All right.
Okay. So yeah, go ahead.
So, I'm unclear. I'm still unclear about why mitochondria fuse with each other. Could you elaborate more?
Yeah. So in context of mitochondria, so the function of mitochondria that are longer has been measured by multiple groups, and they see that the electron transport chain involves moving electrons from one complex to the other, right? That seems to be functioning better when you have longer mitochondria. So as a result, you have better energy production. In other instances, what also happens is rather than doing this mitophagy, you could also, if you have small amount of damage, if that mitochondrion fuses with a longer mitochondrion that is undamaged, you could essentially dilute out the damage in total. So these are two instances. So of course, we are doing these suppositions after having learned about the way the mitochondria function, but evolutionarily, why they arose and all of those are questions I don't think I'm very equipped to answer.
Yeah, I have one more question. Sorry. So when you said that the microtubules depolymerize, then only the mitochondria is left on its own, right? So when when it depolymerizes and those monomers are left, so do they just diffuse out to the rest of the cell, or
the mitochondria?
Is there the monomers?
Oh, the tubulin. Yeah, they do. Yeah.
So do they diffuse out just, or is there an active transport from that site of depolymerization to another site where another site in the cell where polymerization is occurring?
So no, when the microtubules depolymerize, the homodimers that make up, the heterodimers that make up the alpha beta tubulin heterodimers that make up the microtubules just diffuse into the cytoplasm. There isn't a necessity for active movement either in these cells or in other cell types. There are instances of short filaments of microtubules being transported, but that's not in this context. It's a totally different context. But in most cells, diffusion is sufficient. It's a small protein and it can diffuse pretty effectively.
Yeah.
Okay, I have a question. So you said during the cell division, there is more fission and the number of mitochondria increases, and that reduces the partitioning error. So is it just because of the central limit theorem that the partitioning error decreases, or is there some mechanism for deciding which mitochondrion goes where?
No. So it's essentially a binomial process wherein you effectively have a coin toss to determine where you end up. So these smaller fragments, like I mentioned, are able to also diffuse. So long as you have enough numbers of mitochondria and you divide the cell in the center, which these cells do, you should be left with equal amounts of cytoplasm on both sides. And as a result, also mitochondria, which are distributed equally. So the reason that you have a semi-equal distribution of mitochondria is because you're dividing symmetrically at the center of the cell. And also because the cytoplasm is equally distributed with the mitochondria, presumably because these are smaller, they're able to occupy the space in the cytoplasm equally on both sides. So you can imagine that if the mitochondria were bound to the microtubule or there were fewer mitochondria, they would have, there may be instances where you'd have fewer volumes of mitochondria on one side versus the other, just because of the way that the mitochondria are present at a specific point in time. By dividing the mitochondria up and just distributing them isotropically inside the cytoplasm, and by just dividing the cell in the middle, you're ensuring that the cells that result from cell division have almost equal amounts of mitochondria. That's about it.
Okay.
But a related question. So is there enough time during this division for this stochastic or for this division to binomial distribution to even happen? How?
I mean, so if you look at this, you can start seeing that the mitochondria, so let me start playing it again. So this is when the cell is going from having microtubules in the cytoplasm to only having this. And in a 10-minute period, essentially you can have tens of fission events. The rate of fission is about 1 a minute. So each mitochondrion is capable of dividing at that point in time. And that's more than sufficient, because if you look at the total number of mitochondria just before it enters mitosis, it's about 5 or 6. And that's more than sufficient.
Okay. Yeah. So there are a few more questions. If you would like to take them, if you have time.
Let's maybe take a couple more questions. It's okay?
Okay.
Yeah.
Yeah. So Kanishka has been asking about the motor proteins involved in this movement of mitochondria. Is it dynein or kinesin, which is mainly known to carry mitochondria?
Both.
Okay.
That's how you ensure distribution across the entire cell. So if there were more kinesin-driven movement, for instance, you'd have more mitochondria at the cell center, at the nucleus. So that does happen, but that is only when you perturb the system and have more kinesins carry the mitochondria. That's when you'll have it at the cell center, near the nucleus. If you were to perturb kinesins movement of mitochondria, then you'd have it sitting at the cell periphery. So you can change that up, and both motors participate.
So it's a balance of both kinesin and dynesin.
Yeah, kinesin and dynesin. Yeah, exactly.
So another question is, are intermediate filaments also involved in mitochondrial movement?
Movement, no. There are no known motor proteins that bind to intermediate filaments, of course. But dynamics and positioning, possibly. That I'll leave it at that, because there's not a lot of data on how this happens, but there is some evidence to suggest that the positioning of mitochondria is reliant in some cell types on the intermediate filaments, but there's not a lot known.
Okay. So Avni has asked me to read her question now. She's asking, sure, other than polymerization and depolymerization of microtubules, are there any additional intra-extracellular factors that can affect mitochondrial fission?
Right. So, so basically, in this instance, the attachment of mitochondria to microtubules, right, is mediated by a linker protein. So that is, of course, a kind of interaction that you can imagine. The motor proteins carrying the microtubules, carrying mitochondria on the microtubules can also change the way that the mitochondria undergo fission and fusion. You can imagine that two motor proteins are pulling the mitochondria in opposite directions. That is also known to initiate fission of mitochondria. So there are many different things that you can have that changes the way that the mitochondria interacts with microtubules. So this is something we're going to explore in more detail in our future work, because like I mentioned, in fission yeast, it's fairly limited in its capability in terms of understanding this interaction, simply because there's only one known interacting protein, which is this MMB1. In mammalian cells, we anticipate that in addition to probably having linker proteins, which are yet to be identified, motor proteins, of course, are known. It's going to be interesting to understand in a range of different ways in which you could have changes in which the mitochondria undergo fission and fusion based on how the microtubules are.
So one more thing is so this current work that you've showed, the work that you have done about the microtubule length and mitochondrial fission. Could you tell us a bit more about the future work that you just showed? What are the next steps that you are planning?
In terms of fission yeast, we are going to be looking at, we had an interesting finding, which happened, so Liba from the lab, one of the first authors of this study as well. What she noticed was there was a difference in the way the cells divided depending on how the mitochondria looked. And that is very interesting because it seems like there is maybe a two-way feedback between microtubules and mitochondria. So here we show the microtubules change mitochondrial dynamics, but what she's been able to show in preliminary experiments is that the reverse is also true. Mitochondrial attachment to microtubules also changes the way that the microtubules behave. Some people have shown this in the past as well, but we've explored it a little further and we have some interesting results. And that's something we'll hopefully be able to publish soon.
So where does neurodegeneration come in? Is that a direct direct question that you're trying to answer? Or?
Yes, this is the question that we will answer in the future, but I can't tell you any more at this point in time. Yes.
Okay, I think that's it. Yeah, so maybe we can end the session now.
I have a question. So
yeah.
Yeah. So you have been an advocate for women's equality in academia, right? Why do you think that representation of not just women, of any marginalized community important? What difference does it make?
Well, I think first of all, it's a question of being fair, given the fact that women and underrepresented communities make up the population of any given country, women up to 50% and marginalized communities, whatever percent they do, given the fact that they do make up the population of the country, I think it's only fair that they are allowed to, actually not allowed, they have the right to be part of any organization. So that is, of course, the first thing. But time and time again, several studies have shown that bringing in people from marginalized communities, getting diversity in the workforce, changes the way that people work, making it better in every instance that it's been looked at. It brings you different perspectives and gets you to think creatively about problems that have been hard for several centuries. So I think it's only fair to bring in people that are not just the population that is currently in power, but the fact that we make up the population of any country, organization, we need to be, we need to have a seat at the table for sure.
So actually, if you like, I think there would be a few more questions on women in science. If you like, we can talk about it for some time.
Yeah, maybe another 10 minutes. Is that all right? I actually need to get a bit of water for myself as well. I run out of water as well.
Sure.
So if could we take that, if you don't mind, could we take that offline with the participants in this?
Right.
Yes.
Yeah, that's good. We can do that. We can
stop the live stream.
Yeah. Thank you.
Let us all thank the speaker first, to all our live stream audience. Thank you for joining us.